Hey guys, thanks for joining me here on the Come Reason podcast. I just wanted to let you know that if you enjoy these shows, you can gain access to a lot of additional content by becoming a Come Reason Defender. With your tax-deductible gift of $25 a month, you'll be able to view exclusive videos, debates, and presentations, even by top scholars such as Dr. J.P. Moreland, Sean McDowell, J. Warner Wallace, Dr. Hugh Ross, and many, many more. Plus, our Defenders Library provides PowerPoint presentations and notes used to develop these podcasts. So check it out by logging on to the comereason.org/defenders page on the website and see all the benefits your support provides. And thank you for supporting Come Reason Ministries. What does this year's Nobel Prize have to do with intelligent design? Well, a lot more than you may imagine. For years, evolutionists and materialists who study biology have tried to dismiss the theory of intelligent design as not, quote, true science, unquote. However, with more and more discoveries pointing to the intricate and complex ways the machinery inside our cells operate, it's become harder and harder to do so. In fact, this year's Nobel Prize for Medicine or Physiology was awarded to two scientists for their discovery of micro-RNAs that play a key role in the production of proteins of living organisms. Hi everyone, I'm Lenny Esposito from Come Let Us Reason Together, and on this podcast I've invited Dr. Fazale Rana of Reasons to Believe, as well as Dr. Casey Luskin from the Discovery Institute, to review and discuss our Nobel Prize topic for the day, how it supports intelligent design as a model, and how it weakens the evolutionary paradigm. So stay with us as we dive into a fascinating discussion on the mechanisms of life. On this, Come Let Us Reason Together. Well, hello everyone. Welcome again to Come Let Us Reason Together. And as I said, today we are going to be dealing with an idea of the Nobel Prize in Intelligent Design. I mean, it wasn't that long ago that as we were talking about things like ID, the science was actually dismissed as something like creationism dressed up in a cheap tuxedo. And of course, while that line got a lot of play, at the same time, there were some interesting developments happening in molecular biology that led to this year's 2024 Nobel Prize in Physiology. Now, you may not have heard this because, first of all, it's a little bit technical, but also I think the prize was awarded on October 7th, the press being what it is, that was a very famous anniversary date. But let me just, for the audience's sake, recap a little bit about what happened. Doctors Victor Ambrose and Gary Ruvkin discovered these micro-RNAs, which is a new way that we understand how gene expression works. And because I'm not a biologist, I have with me on the show Dr. Fazale Rana of Reasons to Believe, who's an expert in biochemistry, as well as Dr. Casey Luskin of the Discovery Institute. And gentlemen, first of all, welcome. Thank you for joining me here.
Yeah, Lenny, thanks for having us. And Casey, it's always a blast to be able to hang out with you. So looking forward to it.
Same here. It's great to be with both of you guys today, Fuzz and Lenny. Really appreciate the invitation.
Well, and I'm really interested because Casey, actually, I think it was one of the newsletters that the Discovery Institute put out that first kind of brought this to my mind. And as I was reading it, I was much more intrigued. As I said, you have these things where it looks like it's easy to dismiss, but there's a lot of stuff going on. Now, we're going to have to go all the way back to maybe first year college biology to help understand this. So before we get into the details, let's just kind of take a broad overview, because I think a lot of people, when they hear about DNA and replication and things like that, they think about the model very, very simply. They think about it like building a house, let's say, where there's a blueprint, right? And that's the DNA. And they say, oh, well, you just take the pattern from the blueprint and then you go and construct the thing. And that's probably not accurate to say, because I would think that a better analogy may be like an orchestra. where you have the conductor who has a master set of music, right? He's got the master music sheet. But you've got to tell which person to play which part in which pitch and things of that nature. So with DNA and development, you may have heard the ideas of replication, transcription, and translation. Fuzz, can you kind of tease out how this works? Say if you want to build a new protein for a muscle cell or an eye or something.
Right. And before I do that, Lenny, what you're discussing here, though, really represents, I think, one of the grand mysteries of science, and specifically the central problem right now in biology, which is how does the information that is stored in DNA actually translate to the biological characteristics of an organism, right? Which,
yeah.
And so it's in technical terms is how does genotype relate to phenotype? We know that it does, but nobody knows how that relationship or what constitutes that relationship. And in fact, as you're pointing out, there's all these metaphors that are flying around, like, it's a blueprint, or you're using the example of, of a music that's then there's an orchestra and a conductor, using that music to put together a symphony. One of the latest metaphors that's used that I think actually may be hitting on something profound is that the genome, the information in DNA is essentially highly compressed information, and that the process of development is like a generative AI system where that information is being decompressed through the course of development. And when you look at the argument, it actually is a very interesting and very powerful argument. So point is, you're hitting upon the grand mystery of biology, and I think the Nobel Prize that we're talking about is one little piece or one clue to how that mystery may ultimately be solved. But in DNA, the information in DNA is the instructions to make proteins. And proteins are these workhorse molecules inside the cell that carry out all the different activities necessary for life. They play a critical role in forming cell structures. That information that is stored in DNA is segmented, if you will, where the information needed to make a single protein molecule is stored in a region of the DNA that's called a gene. When it's time to produce that protein, the cell's machinery reads that information, produces what's known as a messenger RNA that is a single-stranded version of DNA. That's one way to think about it. And then it migrates to these structures called ribosomes, where the information in the RNA is read and then translated into proteins. And, the information in DNA, again, is like a dictionary, where, in the dictionary, it's all the words that are found in the English language, right? But then when you write a novel, you're not using all the words in the dictionary. Some words you're using quite often, over and over again, but sometimes you're using words only in specific places in the novel, or a single time perhaps, right? And then if you write a different novel, you're using different words from the dictionary, and this is what's happening in the cell during the cell cycle. Some proteins are always needed. These are called the housekeeping proteins and encoded by the housekeeping genes. Others are essentially turned off. Those genes are turned off until that protein is needed. Then there's a mechanism to turn the genes on. The proteins are produced. When those proteins are no longer needed, those genes are turned off, right? And so these are called, inducible genes. And when the genes are inactive, they are referred to as being repressed. And during the course of development, where a single-celled organism grows into a fully formed adult individual, the genes are being turned on and turned off throughout that process. That's what's driving cell specialization and cell migration and secretion of materials in the extracellular space. So the information in DNA is making proteins. Those proteins are produced through what's called the central dogma of molecular biology. But it's a grand mystery in terms of, again, how does the expression of those proteins translate into the phenotypic structures that characterize organisms?
Okay. So yeah, and remembering my biology, basically the DNA, most people are familiar with the double helix structure, right? Like a twisted ladder, if you will, which unzips. And what RNA does is every nucleotide has a match. Every portion of that DNA has a mirror image guy who would match against him. And this is how it's able to send a copy of itself. The mirror image guy comes along and they make sure that they line up appropriately. Then that RNA goes off, that messenger RNA, the DNA closes itself back up, and it goes to the protein building factory, the ribosome, right? And because it has a strip of code and you can only match those elements in a single way, it at least knows how to begin the process. But then there's secondary issues of how the protein folds and as you say, which parts of it are expressed or not expressed, and then there's also a mystery of how it gets used within the system itself. So, but I just kind of want to give a picture again of how, because I know there's a lot of folks who aren't as technical in this regard, just to try and so you have that and then the entire DNA molecule will be reproduced when you reproduce new cells. So you have this concept of, again, you replicate what's in the DNA, you transcribe it, and then you translate it into a protein. So one of the things, and
well, Lenny, could I jump in real quick?
Yeah.
So Fuzz just gave a great explanation of much of the molecular biology that's going on in the cell. And Lenny, what you just described is what is often called the central dogma, which is this very basic fundamental idea of what's going on with information in the cell. The DNA makes RNA, RNA makes protein, and proteins make us. And of course, the central dogma has some truth to it, but we now know that the central dogma is just a very, very gross oversimplification of what's really going on in the cell. That proteins are not the only endpoints of the information in DNA. The information in DNA is also making all kinds of RNAs, and those RNAs, this is part of what this Nobel Prize is about, have all kinds of important functions in the cell. And there's a lot more going on than just, protein-based genes. There's RNA-based genes, and it's not even clear that the entire quote-unquote blueprint of life is just in the cell. I love the orchestra analogy that you gave earlier. I think that that's true. I think that there's so much going on in the cell, and yes, the DNA is a big part of that, but it is just one part of it. There's also all kinds of other components in the cell. There's a cell membrane. There's RNA molecules that are performing functions. There's proteins that can then go back and affect the DNA. And so there's all kinds of this huge orchestra symphony that's going on in the cell, and that ultimately is what does make us. It's not just the central dogma that's important here. Yes, and that's kind of what I've been thinking about as I've been trying to, because it is tough. There's a lot of moving pieces. The AI model is fascinating, Fuzz, but I'm almost scared to say I would need to be able to explain how AI works before I can use that model as a point of clarification. But if you and again, this may break down. I'm still working this out, but as an orchestra, you have the conductor, of course, that not only dictates how the thing should be played and feel, things like that. You've got the individual musicians who have some sway over how the notes are voiced. You have the instrument itself and dampeners and things like that will change the sound of the instruments to make the overall music sound different. And so it's not just the sheet music, or the sheet music does give you direction and all these things, but there's so many other pieces to the music. And one of the things was that again we was fascinating was this micro-RNA was thought to be just kind of
junk material. Now, we've heard the phrase junk DNA before. And matter of fact, Fuzz, I have your book here, Thinking About Evolution, where you touch on pseudogenes. And this has been brought up many times before. The idea was, again, in the 90s, that about 97% of DNA they believed was non-coding. In other words, it was just sitting there from our evolutionary past. That was the assumption. Well, this is DNA, doesn't do anything, so therefore it must be junk. But we now know that that's not true. So can you kind of explain what's happened with this idea of junk DNA and pseudogenes?
You want me to do it, or
yeah, go ahead, Fuzz, and then we'll have Casey jump in.
Yeah. Well, Lenny, you're right. I mean, when even well before the human genome was sequenced, there was this mindset that, as you're saying, most of the genome of organisms was essentially the vestige of an evolutionary history, where there were these different types of sequences that were in the genome that were just the outworking of biochemical processes where sequences of DNA would become physically instantiated within the genome and just remain there, whether they were things like pseudogenes that were genes that were once functional that became disabled, or whether they were, of viral origin, like endogenous retroviruses or other elements that would be derived from endogenous retroviruses, and over the last, 25 years now, since the human genome has been sequenced, there has been really, in my mind, a revolution in how we think about the genome, where there's a growing recognition that, most, if not all, of the sequences in the genome are playing some kind of role. And it's a very interesting debate that's taking place among scientists, independent of the ID creation evolution conversation, about how much of the genome is functional. And that's becomes a very technical debate. It's a very fascinating debate. But you have, a group of scientists who we would say that are working in genomics largely, who would say, no, most of the genome is functional. It's very helpful to think of the genome as functional, to the point where even sequences like endogenous retroviruses that people just dismissed as, again, being the leftover remnants of retroviral infections, now the mindset is, before we dismiss this as non-functional, let's look to see what these sequences may actually be doing. So there's a mindset where now people are looking for function versus simply dismissing sequences as being non-functional. But the group of scientists that tends to challenge the notion that most of the genome is functional are evolutionary biologists. And the primary reason is because if indeed most of the genome is functional, it undermines key tenets of evolutionary theory. I years ago did a debate on Darwin Day with P.Z. Myers at North Dakota State University. Nothing's better than being in North Dakota in February, let me tell you that.
But brutal.
Yeah, but I remember during the debate, P.Z. Myers said to me that we know that the results of the ENCODE project, which has been the primary project that is making these assertions that most of the genome is functional, though other independent studies are affirming it. He said, if the ENCODE project is correct, that it means that ideas in evolutionary theory must be wrong. Therefore, we know that the ENCODE scientists don't really know what they're doing, right? He was very disparaging of ENCODE scientists. And it was really a bizarro world approach to biology, where he was basically saying, look, the data is being evaluated in light of the theory, not as science should be done, where the theory is being evaluated in light of the data. It was, again, this bizarro world idea, but he's not the only person I've heard say that. Time and time again, it's like this can't be right, because if it is right, it means we have a fundamental misunderstanding about evolution. So anyway, that's kind of my quick overview of how the thinking has changed when it comes to the genome and the concept of junk DNA.
Casey, how does this upset the apple cart? Again, if folks don't realize, I don't know if some people even understand the fact that what a virus does, any virus, it's not a bacteria. Bacteria is kind of a self-contained organism, but a virus borrows your DNA machinery, your replication machinery, I should say, and sometimes it borrows it to the point where the virus itself actually gets put in as part of your DNA and becomes part of you forever. And there's a certain subset of those that are, I'm forgetting the Germanic, the germ-based viruses, which means that you can actually pass that virus in your DNA to your progeny, to your children. And that's what we're talking about when we say retroviruses. There was a thought process that, this is something that one of the hominid ancestors of humans had, 800 million years in the past or what have you. And we've just carried it along in our family trees throughout that. But what you're saying, Fuzz, is if this shows to be functional, then it's more like, no, the designer had to use it in all of these options. So Casey, what are you hearing in all of this?
No, you guys have both given very good explanations of sort of the history of this junk DNA concept, how it was born and bred from the evolutionary paradigm, and then really the Human Genome Project in the early 2000s found that there are only about 20,000 genes in the human genome. And biologists said, well, that can't be all that's going on, because that's about the same number of genes that, say, you find in a fruit fly or something like that. So what is making the difference between a human being and a fruit fly if we both have the same number of genes? And this began to be sort of the breakdown of this idea that the only thing that matters in the DNA is the protein coding gene. And so they then began to study what is a lot of the non-protein coding DNA in the genome doing. And this was then in the late 2000s, the early 2010s. Fuzz mentioned the ENCODE project. That's a huge consortium of hundreds of biologists around the world who were studying many of the non-protein coding DNA elements in the genome. And what they found in a huge set of papers they published in 2012 is that over 80% of the genome shows evidence of biochemical functionality. And this was a huge breakthrough against the idea of junk DNA. Basically, that the vast majority of our genome is in fact functional. I love your story about P.Z. Myers, Fuzz. That's a great, very, very nice story, because he's absolutely right. It really was the evolutionary biology community that both created this idea of junk DNA, and then they are still defending it today, even in the wake of huge tidal waves of scientific papers that are now finding function for the non-protein coding DNA. P.Z. Myers called the ENCODE researchers, quote, fundamentally dishonest. Larry Moran, who's another, diehard defender of evolutionary biology, he said, I guess I'll have to be content to point out that many scientists are as stupid as many intelligent design creationists. That was him attacking the scientists who worked on ENCODE. And it wasn't that, he had data to go against what they were saying. He just didn't like the conclusion that the vast majority of the genome is functional. So this Nobel Prize that we're talking about here from Ambrose and Ruvkin, what they basically found is, and they made this discovery back in the 90s, by the way.
Yeah, this is well before ENCODE.
Yeah, I mean, it takes a while for the Nobel Prizes to get awarded, right? Maybe a couple decades. So this was discovered back in the 90s. It's been known for a long time that parts of the DNA that do not encode proteins can have function. And this is what we would call non-coding DNA. These often will encode various types of RNAs. Now, there's many, many different classifications of RNA. Of course, you have the messenger RNA that Fuzz talked about earlier. That's the RNA that does actually go out to the ribosome and help produce the proteins. There are long non-coding RNAs, very long RNA molecules that can have all kinds of metabolic, catalytic, and regulatory functions in the cell. But the particular type of RNA that the Nobel Prize was just awarded for is something called a micro-RNA. These are very short strands of RNA. They may only be a couple of dozen nucleotides in length. But what they found they can do is that these micro-RNAs can go and bind to the messenger RNAs that are supposed to be encoding, the proteins that can then be translated at the ribosome. But when these micro-RNAs bind to these messenger RNAs, they can basically prevent translation and basically regulate whether or not that gene is going to be turned on or off, or how much of that particular protein you're going to produce in the cell. And there are numerous ways that RNAs can actually regulate the production of proteins in the cell. So this gets back then to that central dogma that I mentioned earlier. The central dogma, Lenny, really goes hand in hand with the idea of junk DNA. It's basically that both of them represent a grossly oversimplified view of the genome, where it's largely junk, and it's just sort of this very basic, simplified, oversimplified view, almost going back to the way Darwin thought that the cell was a simple glob of protoplasm. We now know the cell is far more complex than that. Well, the neo-Darwinian view of the genome was this grossly oversimplified view that basically DNA makes RNA, RNA makes protein, and then proteins are like these magic molecules that can go out of the cell and do everything you need. No, there's many, many examples of the DNA encoding RNA, and that's all it's encoding at that point. Those RNAs have all kinds of important functions. And so this is not only showing that the junk DNA paradigm is false, that those non-protein coding sections of DNA can encode RNAs that have important functions, but that it's not just DNA makes RNA makes protein. Sometimes the DNA makes RNA, and that RNA, again, is an endpoint unto itself that has many important regulatory functions in the cell. This shows that not only is junk DNA wrong, but the central dogma is wrong, or at least it's vastly incomplete. There's so much more going on in the cell. Every time we look at the cell, it's more complex than we thought. So we can get into, who predicted this, ID versus Darwin and all that, or creation versus Darwin, or the evolutionary camp. But I think this is really exciting if you're not an evolutionist. We certainly anticipated these discoveries.
Yeah, go ahead, Fuzz.
Yeah, let me just build off of what Casey said. And that is, prior to this discovery of micro-RNAs, the way in which people understood gene regulation, or how you would regulate protein production, would be essentially through transcription factors, which are proteins themselves, that would bind to regions of the DNA that would prevent the cell's machinery from producing RNA transcripts. So the understanding was all the regulation is happening by preventing RNA production, or by allowing that RNA to be produced and then regulating how much of the RNA is produced, where more RNA equates to more proteins. And so what the micro-RNA discovery indicates is that there's another mechanism of regulation that's called post-transcriptional regulation, where after the transcripts are formed, you then you either allow them to be translated or you inhibit their translation, either by breaking down the RNA or somehow frustrating the translation at the ribosome. And so this is a completely different level of gene regulation that was completely unknown to people. And what's interesting is that when you look at the mechanistic aspects of how these micro-RNA molecules are produced, it's a very elaborate production process, and there's multiple pathways. And then once the micro-RNAs are produced, how they are affecting the post-transcriptional regulation, it is an incredibly elaborate, detailed process that is exacting, that is fine-tuned, that is highly precise. And so this is a beautiful example of what I like to call integrated complexity, which is a variant of irreducible complexity, where now instead of the irreducible complexity being manifested at the level of a specific biochemical system, it's essentially a system-wide irreducible complexity, where you've got this highly integrated, highly fine-tuned process to the point where anything that goes slightly wrong in that process is going to foul up the development process. Micro-RNAs play a key role in regulating growth and development, or it's going to lead to certain disease states. It's part of the etiology of diseases. And so this is giving us a real insight into just the elegance and the complexity of how this mechanism works. This is not something that's just slopped together through some kind of evolutionary co-option, but rather these are highly precise systems that bear the signature of design. Just in and of themselves, they bear the signature of design.
Wow. So So it may be, and we don't know all of the ramifications here, but it may be that like my hair used to be fuller than it is now, and it might be micro-RNAs being overreactive that makes me stop producing as much hair as it used to. In fact, we can all blame
micro-RNAs.
Well, and micro-RNAs, they're single-sided, if I remember.
Yeah. Well, I mean, there's somewhere, I've seen differing numbers. Casey, you might know better than me, but there's somewhere between 1,000 to 2,000 distinct microRNA species in the human genome, and it turns out that you've got these very elaborate microRNA networks where there are regions on RNA molecules called microRNA recognition elements, MREs, and so a particular type of microRNA can regulate a large number of genes, and a single gene is regulated by potentially several microRNAs. So this creates this very elaborate network of RNA molecules that include not only messenger RNA, but like the long non-coding RNAs that Casey's talking about, that form this network where where the fate of these RNA molecules in the network is all intertwined with one another, right? And so you've got this incredibly complex, incredibly elaborate system that, again, to me, just bears the hallmarks of design in and of itself.
What's interesting about this, and then Casey, I'll let you comment, is one of the areas that I have background in is information technology. And if you're building a database, you can actually store everything in one table, but your table gets very large and very difficult to manage. You can try to store things in a couple of tables and have a one-to-one relationship. But if you have a many-to-many relationship, it's a much more compact way of storing the information, much more efficient as well. So what you're saying is this is actually, from an information standpoint, a very, very efficient way. Because you had said, only, I think, Casey, 20,000 genes that the DNA strand has. How do you express it in this myriad of ways? And that's what we start to see here. Is we start to see a level of complexity, but also it brings a level of efficiency to the entire project. Is that fair?
Yeah, I mean, what you and Fuzz are talking about is really this orchestra, Lenny, that you mentioned. You have
That's
all
right.
these different players that are involved. As Fuzz mentioned, there's different species of RNA. We've got microRNAs. We've got long non-coding RNAs. We've got messenger RNAs. There's other kinds of RNAs we're not even getting into here. And they interact with proteins. They can regulate the production of proteins. Proteins can regulate the production of RNAs, and they can even go back and change what reads in the DNA sequence of the genome in some cases with those retrotransposons and these genetic elements that can move around the genome, literally reinsert themselves and provide new functions. So yeah, there's this huge orchestra of different molecules in our cells, and together that's what makes us. And I think that this is exactly what would have been anticipated by an intelligent design kind of view, that the more we look at information, the deeper we look into the cell, the more we find information, regulation, and control. And so I'm seeing sort of four basic levels of information that we're talking about here. And it's far more complicated than this, but what are those four levels? Well, first, we've got information in the DNA that encodes proteins. Second, we've got information in the DNA that is not encoding proteins. This is the non-coding DNA that our evolutionist friends used to say was, the 95% or so junk of the genome, whatever, they give different numbers. But, so we got the non-coding DNA that is producing RNAs. That contains information. Then there's information that's what we call epigenetic information. And we haven't even talked about this yet, but this is where you're tagging genes, tagging the DNA molecule with different tiny molecules. They could be methylated. They could be acetylated. There's all kinds of different ways you can have different molecules in the cell that are latching onto the DNA, and those can turn genes on and off. But those epigenetic controls are still connected to the genome. And then the fourth level is we even have information in biology that really is disconnected from the genome. Information that might be carried in the form of sugar molecules on the cell membranes or electric fields that are generated by organisms as they're developing as an embryo. And that helps to guide, which cells go where. Or within a cell, you have these microtubules that can tell the cell where to take different molecules and different protein packages and so forth, and how that cell should be operating. And that is not necessarily encoded in the genome. That can be encoded in the parent cell that produced that daughter cell. So there's all this information in the genome at different levels, and we're only really right now talking about the protein coding DNA and the non-protein coding DNA. But there's even more beyond what we're talking about here. And again, this is the orchestra, and it's far more beautiful and elegant and complex. I just read the book, How Life Works by Philip Ball, and he keeps talking about how biology is dizzyingly complex. And I love that phrase. He's an evolutionist, of course, but I think from a design standpoint, we're not afraid of going there. Whereas the reductionist, materialist folks, they're the ones who are always pushing back on the idea that the cell is more complex than we previously thought it was.
Well, and we're probably,
getting a little deeper into the weeds, but that's okay. Yeah.
Yes, and we're at the danger of getting far afield here. Casey was mentioning this idea that the central dogma isn't really incorrect; it's probably incomplete and highly simplified. And I'm very much in that camp. But micro-RNAs actually are leading to another companion idea to the central dogma that I think is going to be one of the most profound ideas in biochemistry, and that is called the competitive endogenous RNA hypothesis, which is a mouthful. But in effect, micro-RNAs are at the center of this idea. And the idea here, and this idea actually explains the functionality, at least in part, of pseudogenes in the genome. Because pseudogenes are believed to be this evolutionary byproduct of what was once functional genes. And if I'm not mistaken, about 2% of the human genome consists of genes and that are encoding proteins, and about 2% are considered to be pseudogenes, if I'm not mistaken. So there's almost as many pseudogenes as there are genes in the human genome. But it turns out that because these pseudogenes oftentimes are transcribed, they become part of this population of RNA molecules that Casey was talking about. So it's not only messenger RNAs, but it would also be the long non-coding RNAs, the micro-RNAs, but also it would be the transcripts from pseudogenes. But these pseudogenes transcripts are going to have these MREs, the microRNA elements with them, which means that they now become part of this network of RNA molecules that are being transcribed, where RNA molecules that share the same MREs are part of a family. And so what happens is that if you upregulate one of the members of that family where you're producing more RNA, what ends up happening is that in effect, all the other RNA molecules that are in that family, those that are coding for proteins are going to be transcribed at higher levels, right? And so what happens is, like with duplicated pseudogenes, these are believed to be genes or pseudogenes that were generated by some kind of duplication event that made a copy of the intact original gene, and then it underwent mutational change because a second gene is superfluous, right? And so that it still resides in the genome as a duplicated pseudogene. Well, there have been these classic studies that have taken place where people have shown that if you essentially prevent the transcription of the pseudogene, it actually leads to a disease state. And one of the first experiments was involving a protein called P10, which is a tumor suppressor. And so if you disabled and prevented the P10 pseudogene from being transcribed, you'd wind up with high levels of cancer in the mice. right, that were being studied as the model system. If you allowed that the PTEN pseudogene to be transcribed, tumor suppression took place. And what ends up happening is that duplicated pseudogene is serving as a sponge that's binding up a significant portion of the microRNA, which is now allowing the PTEN gene to be translated and produce the protein that plays a role in tumor suppression. Now let's multiply that effect across an entire network of RNA molecules that are forming a family. And so in this case, the transcribed pseudogenes are actually in this network playing a key role in maintaining some kind of homeostasis with regard to the RNA species, and are playing a role in consuming up microRNAs, allowing critical messenger RNAs to be translated, right? And so this also explains why unitary pseudogenes are necessary in genomes. It's because they too are playing a role in these microRNA organized networks.
That's really interesting. So basically what you're saying is there's a gene that does a job, and then there's for lack of a better word, a decoy. And the microRNA is an inhibitor. It's a thing that stops the gene from doing what it's supposed to do. So what they do is the body sends out the decoy to attract these things, and all of them bind to the decoy. That allows the gene to express itself, and now we have more cancer inhibition or something to that effect.
Yeah. And one of the big conundrums is that, how do you explain unitary pseudogenes? Because they're just a single copy of a gene that's been disabled. Well, there's an elegant explanation now for why unitary pseudogenes are in genomes. It is because they too are being transcribed, or at least some of them are, and those transcripts are entering into these networks, and to remove them now alters the homeostasis of these RNA networks, which is to the detriment of the cell, right? So so this also now allows us to explain not only that pseudogenes duplicated and unitary are functional, but it also goes one step further and explains why these sequences look similar to genes, right? There's a reason they look similar to genes, because that similarity is central to the way they're functioning. So that if I was a designer wanting to create, some kind of regulatory mechanism, I would introduce these elements into the genome that look like genes, but in fact were not. Right.
And if I could chime in here, what Fuzz is talking about right now, I'm so glad that you raised pseudogenes, Fuzz, because this is actually one of the most common arguments you will hear from our evolutionist friends. Even theistic evolutionists will say, well, what about pseudogenes? Why do we have these things in our genome that look like genes, but they don't produce a protein? Isn't that much better explained as a gene that was once functional, it incurred some kind of mutation, and it was basically subfunctionalized, or it was rendered non-functional? This is evidence of an evolutionary history and not a design, aspect of the genome. And what Fuzz is saying right now is that no, there are very good functional reasons why these pseudogenes need to look like genes. And what this really highlights is the fundamentally different approaches between an ID-based version of doing biology versus an evolutionist or reductionist materialist way of doing biology, where we look at biology from an ID perspective and we say, you know what, we may not fully understand how this works or what's going on right now, but let's not assume that just because we don't understand what it's doing, that therefore it's non-functional junk, and it's just there because of random mutations loading our genomes up with evolutionary debris. No, let's see if maybe there's a reason for why things are the way they are. Whereas if you're an evolutionist or a materialist, you look at these things, you say, I don't understand what these things do, therefore they must be evolutionary junk. I don't see them doing anything, therefore they're just evolutionary debris and junk, and I'm going to write them off. And now it's turning out that the ID-based view really was the right way to approach the genome. In fact, there was an article in, I think, Nature Reviews Genetics a couple years ago that talked about how we have sort of this dogma and this sort of premature, basically dismissing these pseudogenes as if they are function. And this dogma of that they are just, evolutionary junk is preventing us from understanding what they really are doing. So I think that's important to appreciate that these different approaches to biology really do have practical implications for, what we'll discover and whether biology is going to make discoveries.
I'd like to build off of the point Casey made. Sorry, Lenny. Because Casey's making a really, really important point here. And so often I hear this criticism that, look, if you embrace creationism or you embrace intelligent design, that you're saying that there's an intelligent agent that's responsible for life, or there's a creator responsible for life. It's a science showstopper. It brings everything to a grinding halt. Well, I would actually argue when it comes to this whole idea of junk DNA and how we understand genomes, their structure and their function, it's been the evolutionary paradigm that was the science showstopper. Because as Casey said, when people saw something that didn't make sense or didn't look like it was functional, they wrote it off as just a vestige of evolution. Let's not even bother studying it. And as Casey said, it's people from an ID perspective, from a creation perspective, that would say, wait a minute, if it's there, and particularly if it's conserved, it's got to be doing something, right? It's got to be functional. We may not understand, but it motivates us to press into it to try to understand what the function is. And it's only been really accidentally that many people that hold to an evolutionary perspective have discovered function for different classes of junk DNA, and they publish the stuff, dragging their feet and kicking and screaming because it's contrary to the paradigm. But when enough people start to publish this kind of work, suddenly it changes the mindset, as Casey said, to the point where people are now asking, well, what are pseudogenes doing? And have we actually missed critical functions? What are ERVs doing? Have we actually missed critical functions? And when we start to look for functions, lo and behold, we find function.
Yeah, exactly. Well, we saw the same thing, if you think about it, even with vestigial organs, that's quote unquote, where, people, your appendix isn't something you need, it's just something left over. And then they're finding out, no, it plays a role in the lymphatic system, plays a role in your immune system. If you can keep it, because it actually does do things. And so, yeah, you see that expressed definitely, at least at that level, where people have a stake in it, right? You could say it's not worth anything, but I don't necessarily want the surgery to take it out just because, So I do have another question, though, and this is the other aspect of micro-RNA that fascinated me, the regulation of gene expression, because it seems that and I don't know, correct me if I'm wrong really quickly, does microRNA only suppress gene expression, or are there microRNAs that do other things like fold, or do we know? Maybe we just not have insight into it yet. But whenever I've read up microRNA, it's always been about gene suppression.
That's my understanding, but yeah. I don't know.
Okay. So
that's my understanding, but every time you think you understand it, there's always more going on.
That's right. Okay. But here's my question, because it was fascinating to me. The Evo-Devo model. Let's take an example like maybe the peppered moth, which has always been something that everyone seems to be familiar with. The idea that you have dark melanin moths and light moths, okay? And regardless of, I understand that the latest studies on this have shown that the light-colored moths actually peaked in their population even during the beginning of the Industrial Revolution, where coal was all over the trees, right? And the dark moths, anyway. Regardless of that explanation, there's always been the idea that, well, it was a genetic error that created, say, the light melanin. It was just a miscoding or something like that, and that's why we get things like blue eyes, or that's why we get things like white colored moths. So you have dark colored moths and you have white colored moths, and in the evolutionary paradigm, it's then just survival of the fittest. So there's a happenstance, there's a fortunate accident, right? And then the selfish gene takes over and wants to reproduce itself, and then based on whichever mistake in the genome produces more survival, then that's better. But with microRNA, what I'm understanding is it may simply be that you have two versions of that gene, or you have that gene is able to be expressed in two ways, say light skin or dark skin. And so what I'm saying is it's not necessarily a mistake that some of these plans, these options in body plans exist. It's the fact that you have multiple ways of expressing it to have different ends, right? The instructions are all there, and then something happens, be it an external force, a sugar or something like that, that causes plan A to be expressed and plan B to not be expressed. So this is why I'm really fascinated about this, because now it takes the whole evolutionary paradigm down even another level and say you're not even getting mistakes in the genome to start expressing things. There may be more than one instruction set there.
Yeah, and this is not just necessarily mistakes, Lenny. This can be exactly the way that biology is supposed to operate. In fact, the press release about this Nobel Prize we're discussing talks about how turning certain genes on and off in different cell lineages as an organism is developing, that's what causes an organism to develop into the exact, body shape that it becomes, because certain cells will use these microRNAs to turn certain genes off and leave other ones on, and then they turn into a bone cell. And then in another cell lineage, you're turning a different set of genes on and off, and that develops into a blood cell. And then in a different lineage of cells, you're turning a different set of genes on and off through these microRNAs and other systems, and that becomes, say, a tissue cell or a skin cell or something like that. So it's the turning on and off of genes that really determines what proteins are being expressed in a cell, the proteome of the cell, and that determines what kind of a cell that cell becomes and what it does. And, and that is what builds an organism, basically. So this is important not just for, the way evolution might proceed by turning things on and off, but just for normal organismal development and how organisms work.
Yeah, but in terms of, what the implications are for the evolutionary paradigm, every time you make things more complex, and not just complex, but as I mentioned, this idea of an integrated complexity, the harder it becomes to evolve that system, right, in a productive way. You can evolve it, but it's almost going to always create some kind of hopeful monster. And, and as case in point, we understand now that not only are microRNAs playing a critical role in the process of development by controlling gene expression, but that when things go awry, mutations in microRNA or the machinery that's manipulating microRNA leads to disease conditions, including part of the etiology of cancer. So for that reason, it really makes you wonder how much can you actually begin to alter microRNA biochemistry in support of large-scale evolutionary changes, that would drive, these, or be driven by these large-scale phenotypic changes. It almost seems to me, at least intuitively, that this creates even greater constraint on what's possible in terms of evolutionary transformation. And I would be curious, Casey's thoughts on this.
No, I agree with you 100%, Fuzz. I mean, these gene regulation mechanisms are very complex, and you have to have multiple components there in order for them to work. And what's amazing, that's obviously the kind of integrated or irreducible complexity that Fuzz is talking about. If you don't have all these parts present there, they're not going to work. But what's also amazing is that they're basically using logic. We're literally seeing if-then statements in terms, not actual written if-then statements, but there's a logic to them. So if this microRNA is present, then you turn the gene off. There's many, many different ways that a gene can be regulated. Talk about something like the lac operon, which is found in bacteria, where basically if you don't have lactose, you have this repressor molecule that binds to the chromosome and prevents the RNA polymerase from going along and transcribing all of these genes that are needed to metabolize lactose. And then when lactose is present, that causes the repressor molecule to release, And maybe I said when the lactose is absent, there's a repressor molecule. I think I said that backwards. And then when the lactose is present, the repressor releases from the DNA, and then that allows the RNA polymerase to go along the chromosome, transcribe all of those genes, and you get the enzymes you need to metabolize lactose. Okay, that's an if-then statement. If lactose is absent, the repressor binds to the chromosome, and then you don't get any transcription. If lactose is present, the repressor releases from the chromosome, the RNA, and then the RNA polymerase transcribes the genes, and you get the enzymes needed to transcribe to metabolize lactose. So we're seeing logic circuits. And to go one more level here, what's amazing is we see the same logical sort of pathways being reused in gene regulation system after gene regulation system after gene regulation system throughout the cell. So what's going on here? Is this like convergent evolution times 1,000, of these irreducible integrated complexity, logic circuits? I mean, that's really what's going on in the cell. If there was ever a case for design, where you have systems that require multiple parts to work, they're using pure logic in order to work, and those systems are being reused over and over and over again in different systems across biology, to me, that's just an incredibly strong case for design at many different levels.
Well, to build off your point, Casey, that's exactly what designers do. Designers characteristically produce irreducibly complex systems, integratively complex systems, and employ logic circuits. And if a design works, it's reasonable to use it over and over again. If you think about, these designs as emanating from the mind of a creator, it doesn't mean that God is lacking imagination, as I've heard some skeptics say, but rather that you're looking at these incredibly optimized systems that why wouldn't you reuse them over and over again because of the high level of optimization that you see? So what Casey's describing is exactly the type of thing that designers do over and over again. And in fact, when we see designers produce these kind of systems, we celebrate them. We recognize them as being, brilliant designers that are clever and creative in what they're doing. And yet, for some reason, when we see systems that are even more elegant than anything that a human being could produce, but yet shows that same hallmark, those same hallmark features, we just simply dismiss it as being, cobbled together by evolutionary processes. It's right. So there's just such inconsistency in how we respond to design systems.
You have to work very hard to convince yourself that materialism is true. And just one last little prediction here. Fuzz just gave us some great predictive ways that intelligent design, a design-based view can help us think through what's going on in the cell. But at a very, very basic level, what do designers do? They make things that have a function. Well, that's where the whole junk DNA evolutionary mindset went wrong. They looked at the entire genome and said, well, 90+% of this, depending on who you ask, anywhere from 50 to 95% of the genome is useless junk. And ID proponents and folks in the creationist community the old Earth creationist community that Fuzz works with, we've been saying for a long time, no, we don't think it's going to turn out to be junk. We think it's going to turn out to be functional. And in fact, I remember when I was an undergraduate at UC San Diego, I had Fuzz there to speak, and Fuzz was talking about this, how, the junk DNA would turn out to be functional. That was like 20 plus years ago, before we had this revolution in junk DNA. So ID theorists have been saying this for a long time, that we would find function for the non-coding, quote unquote, junk DNA, and lo and behold, that prediction turned out to be right. It's a very, very basic prediction, but it shows, which view of the genome is really the right one that's helping us to better understand how things work.
Yeah, that's one of the points is, right, ID was dismissed as not science because it didn't, quote, make any predictions. And what we're finding out is, it actually did, and we've started to see them come to fruition, which and Fuzz, you had talked about the reticence of evolutionary biologists to accept things like the ENCODE project and things like that. So my kind of last question as we begin to wrap up is, this is the Nobel Prize. This is not a small piece of recognition for scientific excellence. it's going to be very, very hard to dismiss this as just, a hopeful ideology. It becomes really difficult to. Now, it's not to say that the Nobel is some, imprimatur of righteousness. I mean, the man who invented the lobotomy got the Nobel Prize in 1934, so we have to be a little careful in how we approach those things. But it is to say you can't say that it's outside of proper science. And we actually don't have, even today, there are all kinds of implications for this. If we can figure out a little bit more about how it works, that we could maybe inhibit runaway genes, or maybe somebody is missing, it's not the gene expression that's the problem. They're missing the micro-RNA portion, and that's why they suffer from this malady. We just don't know yet. The medical implications of this discovery are just astronomical, and as scientists continue to look through it, I think that in the future we'll probably see some real practical outcomes for this. But what does this prize, the fact that this is noted, what does this do to the conversation between those who say it has to be non-purposeful, right? Random events that bring about these kinds of developments within the cell. How does a finding like this maybe change the discussion a little bit?
Well, I mean, you're making a powerful point, Lenny, and I like the point a lot. And it's not that uncommon for Nobel Prizes to be awarded in medicine and physiology, chemistry, or in physics, where the implications of those the scientific discoveries that are the basis for the prize have positive implications for the case for design, or for the case, for a creation model. So this, believe it or not, is not that surprising to me that this would have such broad-reaching design implications. And I think what that's saying is that really the best science that is transformative in terms of how we understand the universe or how we understand life is producing the results that we would exactly expect from a design standpoint, from a creation model standpoint. What we like to say at Reasons to Believe, the more we know about science, the more we have reasons to believe. And this is just, another example of it. But, again, the fact that this is a Nobel laureate, Nobel award-winning type of breakthrough, at least in terms of engaging the public at large, is significant.
Yeah, I mean, I totally agree with what Fuzz is saying, and I need to actually write a blog post about this now that I'm realizing just how important this is. I mean, literally the Nobel Prize in Physiology or Medicine this year was given for a discovery that helped overturn junk DNA. I mean, that is what this Nobel Prize was for. And in the press release for this Nobel Prize, they even talk about how at first their discovery was thought to either, not be that important. It was like one little isolated example in one, C. elegans roundworm, and it's not a widespread thing. And now we know that micro-RNA gene regulation is everywhere. And so I think it's important to appreciate that, yeah, just as Fuzz said, this is a discovery that has huge implications for intelligent design. And in this case, I think it's because it's finding, A, that there's function where evolution did not anticipate it, and B, there's complexity where intelligent design would have and did, in fact, anticipate it. And so we can multiply this out by, thousands of times, because you read just about, pick a random scientific paper from a scientific journal, and you're finding discoveries like this that are finding new forms of complexity, new types of functions. This is happening all the time. These two guys did great work. They're getting a well-deserved Nobel Prize from it, but it's not like this is an unusual thing that is happening in biology. And it's because of this that about a year ago, there was an article in Bioessays that says that biology has literally undergone what they called a Kuhnian paradigm shift away from the idea of junk DNA. And this really, I think, is something that was anticipated by intelligent design. There's also a book by John Maddock and Paulo Amaral that came out last year called RNA, the epicenter of genetic information, published by a mainstream scientific publisher, Taylor and Francis. And I just want to read a quote to you from that book. They say, while the story is still unfolding, we conclude that the genomes of humans and other complex organisms are not full of junk, but rather are highly compact information suites that are largely devoted to the specification of regulatory RNAs. These RNAs drive the trajectories of differentiation and development, underpin brain function, and convey transgenerational memory of experience, much of it contrary to long-held conceptions of genetic programming and the dogmas of evolutionary theory. So this is basically a quote from a mainstream scientific book published by mainstream researchers saying that this is discovery, that the genome is not largely junk, is contra to the dogmas of evolutionary theory. And this is exactly what intelligent design would expect. Now, these authors I just quoted, they're not ID proponents. But so why are they saying this? Because they're just looking at what the data is saying. They don't have an ideological axe to grind. They're just saying, look, we know from what biology is discovering that the genome is largely functional and it's far more complicated than we originally thought. And from the corner of the ID camp that Fuzz and I are in, we're cheering them on the whole way. And we have researchers, of course, in our community that are studying some of these same things and making these same kinds of discoveries. So we say, may the research continue and may these discoveries just continue to confirm the predictions of ID.
Well, yeah, that's thanks for that, Casey. So when I saw it, I just noticed some of these implications, and I thought that they were pretty significant in that regard. Any last thoughts, Fuzz and Casey?
No, but I appreciate, Lenny, the fact that you invited us on the program to talk about this, and you have an impressive insight into molecular biology. So by no means sell yourself short. I've been impressed with
thanks.
The insight that you have in terms of the questions and the thoughts that you've shared with us. So it's been fun for me. I always enjoy hanging out with Casey.
Yeah, same here. The feeling is mutual. I really always enjoy conversing with you, Fuzz. And Lenny, yeah, you've brought up a great topic here that I think is more important than actually I appreciated before I prepped for this interview. I'm going to have to go out and do some end-of-the-year blog post about the importance of this Nobel Prize. I think this is very significant. I appreciate you raising this very much.
Oh, great. Glad to hear. Just one other question. Does this have any implication for the RNA World Hypothesis? Does micro-RNAs have any bearing on whether that makes that more or less likely?
That's a good question. I'd have to think about that for a minute. I mean, what it is showing, and this relates to the John Maddox quote, is that RNA biochemistry is far richer than we thought. So that could actually be something that an RNA World researcher might consider a feather in the cap of the theory, but I don't think that, micro-RNAs do anything to fundamentally address some of what I consider the fatal flaws of the RNA world.
Yeah.
But interesting question.
Yeah, I don't see any direct implications here either, but I will say this, that one of the arguments you get for the RNA world, the idea that, the first life was using just RNA instead of DNA and proteins, is they will say, we see vestiges of the RNA world in modern biology's usage of RNA, and they'll typically cite, messenger RNA as sort of just this ferrying molecule that takes information from DNA to the ribosome. Well, now that we know that RNA is not just sort of this, has one little, it's an important function, but one little, RNA is literally, in some ways, it's almost more important than proteins. It's literally running the show and regulating so much of what's going on in the cell. I don't think it's fair to say that RNA is a quote-unquote vestige. I mean, RNA is crucially important to so many aspects of biology. So again, this is just sort of that evolutionary junk mindset saying, this is an evolutionary holdover reflective of this old ancient RNA world day of biology, when in reality, RNA is crucially important. And that's just, again, reflective of their mindset. So we've got to let the data speak for itself. And I think that when we do that, it's not taking us in the evolutionary direction.
All right, guys. Well, thank you so much. Appreciate all your time and love the discussion. And we'll have you on again soon, I'm sure.
Thanks.
All right.
Thank you.
Thank you for watching this Come Reason video. I hope you enjoyed it. If you'd like to see more videos like these, consider subscribing to our Come Reason YouTube channel by clicking on the subscribe button. And you can follow us on social media. Lastly, if you'd like to help keep these kinds of videos free, consider providing a donation by clicking on the donation button beside me. Thank you.